So good morning, class. Welcome to our today's sessions. Welcome to our today's session. We are in our nutrition and biochemical techniques class, and our topic today that we are looking at is demonstration of knowledge of molecular genetics. We want to see how you will be able to acquaint yourself with understanding of the matters, what our nucleus contains. Are we together, class? Okay, so let's go on, and these are the training objectives that we have. We have 4 training objectives. The first one is that at the end of this class, you should be able to define the term nucleic acids, and then secondly, you should be able to identify the 2 types of nucleic acids. I expect you also that at the end of this class, you should be able to distinguish between the various bases that make up nucleic acids. And lastly, can you read that? Yes. Yes. Okay, that's correct. So let's, without taking much or wasting much time, let's look at the introduction into what nucleic acids are. And we begin by definition of the term nucleic acid. What is nucleic acid from what you are seeing there? Yes, nucleic acids are molecules in the nucleus of our cells that store genetic information and control the physiological functioning of the cell, which includes growth and reproduction. That is a very important function of the nucleic acid. Nucleic acids also carry information about or controlling the being able to transmit information from the parent cell to the daughter cell or to the offspring. And we have 2 kinds of nucleic acids. There are 2 types of nucleic acids. The first nucleic acid there you can see is deoxyribonucleic acid, which is usually abbreviated as DNA, and ribonucleic acid, which is usually abbreviated as RNA. Good. So we have these 2 types. These are the only types of nucleic acids that we have. We either have DNA or RNA. But for the sake of in humans, in the context of humans and animals and trees, we don't talk about RNAs. We talk about them having DNA. RNAs may be found in certain microorganisms like viruses and bacteria. But for human beings and other animals and plants, we are talking about DNA. Now, let's look at some of the items that make up these nucleic acids. These nucleic acids are made up of what we call, it's a polymer. And nucleic acids are polymers made up of a linear array of monomers, which are called nucleotides. And nucleotides are made up of 3 components. We have nitrogenous heterocyclic base, we have a pentose sugar, and you also have a phosphate residue. These are the 3 items or the 3 components, the 3 molecules that make up a nucleic acid. And you can see the structure there. Do you look at that diagram? Yes, there is a diagram there showing the phosphate. Good. Yes, we have the phosphate, we have the purine, and you have the pyrimidine base, and lastly the pentose sugar. Very good. So we have a sugar, we have a base, and we have a phosphate group. Now we have 5 types of nitrogenous bases, and we have them categorized into 2 groups. The nitrogenous bases are grouped into pyrimidine and purine. And in the category of pyrimidine, we have the uracil, we have thymine, we have cytosine, and these are the 3 bases that make up the pyrimidine group. What about the purine group? Did it for me? Good. Very good. So we have only those two. Now, class, do you notice any difference between the two groups of nitrogenous bases? Look at the difference between the purines and the pyrimidines. What difference do you notice? Yes. Yes. The pyrimidines are made up of single rings. They are made up of single nitrogenous rings, and we call them nitrogenous bases. By the way, let me mention this one. We call these molecules nitrogenous bases because they contain nitrogen. Like you see the molecular formula down there for uracil. You see it is C4, H4, O2, and N2. So there is a nitrogen there. That is what makes them to be called the nitrogenous bases. So you look at all of them, they have nitrogen. They must have nitrogen. They have at least 2 nitrogen atoms. Do you see that? Some, like cytosine, have 3 nitrogen atoms. Some, like cytosine, have 3 nitrogen atoms, and adenine and guanine have each 5 nitrogen atoms. So they are grouped into 2 like that. And then you've also noticed the difference that the pyrimides are made up of single nitrogenous rings, while the purines are made up of double nitrogenous rings. Now, what brings the difference between DNA and RNA in the types of sugars that they have? We have 2 types of sugars that make up the nucleic acids. We have can you read them for me? Class? Yes, we have the ribose sugar. Yes, very good. We have the ribose sugar and the deoxyribose sugar. These are the sugars that bring one of the differences between DNAs and RNAs. RNAs have ribose sugar, while DNAs have deoxyribose sugar. And the difference between these sugars is that in ribose sugar and the deoxyribose sugar, we consider in that the cyclic structure of that sugar, we look at carbon number 2. We compare the molecules or the atoms that make up the attachments to the carbon number 2, which we call the beta carbon. First carbon is called the alpha carbon, and the second carbon in these structures are called the beta carbon. Do you notice the difference between the second carbons of each of them? Do you see that? What is the difference, class, of ribose sugar? Yes. 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 So in this, very good. Now in this cyclic structure, you realize that in the ribose sugar, we have a H and an OH group, while in the deoxyribose, the 2 attachments in the beta carbon are H and H. So this one, the ribose sugar exceeds the deoxyribose sugar in beta carbon number 2 by having an extra atom, which is oxygen. So these are the 2 types of sugars that make up the nucleic acids. So a nucleic acid, which is a nucleotide, could either be having a ribose sugar or a deoxyribose sugar. Are we together, class? Are we together? You hear me? There is a lot of noise in the background. Okay. So another difference, let me mention this one I have not indicated here, but it's good for you to note that we have the ribose and the deoxyribose sugar. One of the other differences is that apart from having the differences in the structures of their sugars, the other one being ribose and the other one being deoxyribose sugar, we are having is that the DNAs usually have double helical structure, while the ribonucleic acid, which is the RNA, has a single strand of nucleic acid. That is a very important concept. But there is, we shall come to see that there is a special type of RNA which has a double strand, and we shall come to see that one in our next class. We shall look at the very special RNA molecule. Now, as we wind up this class, I don't know if you have any question. You have any question? You've understood everything. Thank you very much. So thank you very much. I want to give you a takeaway assignment, and your takeaway assignment that I want you to go with and complete before we meet in our next class is that you should be able to discuss at least 5 functions of each of these, the DNA and the RNA. You discuss the 5 functions of DNA and RNA. 5 functions of DNA and 5 functions of RNA. Are we clear? Are we clear? Yes. So let's meet in our next class as you do your serious studies and revisions, as we see that our term is approaching to the end, as you prepare for your end of term examinations. Thank you very much, class, and have a very wonderful day.